Well, hello, this is Miss Dagenford, and we are going to be looking at some organic chemistry tonight. We're going to be looking at carbohydrates and then lipids. And ultimately, what you should be able to do is recognize each of these and then understand and predict their behavior, the chemical behavior. That's going to help us throughout the year. Here we have our picture of carbohydrates, and you'll notice that these are predominantly our fruits. Here we have some apples and pears and strawberries and also starches. So we've got some sugars and then starches. Here's our potatoes. Here's our corn. They don't picture grain, but grain like wheat and rice would also be part of that. Now, just a quick note of review. If I sneak over here, let's just make sure we review polarity. And polarity is, remember, going to exist in a molecule where we have atoms that are spatially asymmetric, and also they have different electronegativities. And so our classic example was water, and water is going to have oxygen and hydrogen, which have different electronegativities. Oxygen is much more electronegative, and that will pull the electrons toward it, giving oxygen a partial negative charge. The hydrogens have a partial positive. Our nitrogen is going to have a similar situation. Nitrogen has a partial negative, hydrogens have a partial positive. So as we're going through our organic molecules, keep that in the back of your mind, and we're going to be noticing that. So let's go ahead and start with carbohydrates. Now, carbohydrates have a unique ratio that contributes to its behavior. It's always going to have a 2:1 ratio of hydrogen to oxygen. Now for monosaccharides, it's going to be multiples of CH2O, but that's not always going to be the case. Think carbohydrates, carbon, and then hydrate is water, H2O. But all carbohydrates have 2:1 ratio of hydrogen to water, hydrogen to oxygen. That's always going to be true. So let's take a look at monosaccharides. Monosaccharides, we can see them in linear or ring form, but most often we're going to see ring. So we'll start with trioses, which are 3 carbon. And then we have pentoses, which are 5 carbon. These are things like deoxyribose and ribose, which are found in DNA and RNA respectively. And then we have our 6 carbon hexoses, glucose, galactose, and fructose. Now, one thing that you will notice that these are all isomers of each other. They have the same molecular formula, but they're built slightly differently, and that's going to be enough to change sometimes their behavior. So let's take a look at these structures. Now, trioses, you'll see 3 carbons here. Now, one quick note, and again, keep that in the back of your mind, what I said. Here are the hydroxyl groups, and these hydroxyl groups are going to influence its behavior because of their polarity. So in the back of your mind, you should be thinking, oh yes, I know what that is, and we're going to test you in just a second. Now, pentoses and hexoses can form rings, but trioses can't, because carbon can't exist in these triangular ring forms. That's just not going to happen. It stresses the bonds out way too much, so that's not going to happen. But notice down here, these linear carbohydrates can bend around and then form bonds right here to then build the ring structures. Now, one quick word about the structures. Whenever you see in organic chemistry these angles that don't really have anything there, those are carbons. But notice how cluttered my picture is right here, and you can understand why they break it down into that substitution. There's the angles. Now let's real quickly look at our polarity. Because oxygen is going to have that partial negative charge, let me go ahead and highlight. Here's all of our partial negative charges. Even this oxygen is going to have an ever so slight one. That's not going to be a big one though. And then our hydrogens, now bear in mind this is only going to be the hydrogens that are attached to an oxygen. That's where we see the biggest difference. That's where our partial negative and partial positives are. So let's real quickly go ahead and put that in. Partial negative, partial positive. Now if we were to go ahead and draw water, now we can put things together as to why sugars interact. Here would be our partial negative, here would be our partial positive, and because of that there's going to be an attraction, and that, remember, explains why sugar dissolves in water. This molecule won't break down, but it will separate from other glucose molecules and it will dissolve. Now, what should you know? You're not going to have to draw these, and you wouldn't have to know the difference between glucose and galactose. But you should be able to recognize, yes, that's a monosaccharide, and I know what it does. It mixes with water, it forms these polar bonds, and it forms these attractions, which you're going to have a question about right now. Now, what are their jobs? Well, their jobs are to provide energy, and then they're going to be building blocks to build other carbohydrates and glycoproteins that we can connect them together with other molecules. Disaccharides, well, disaccharides come about from two monosaccharides. We join them together, we form a glycosidic linkage or a glycosidic bond, and we're going to do so through a process called dehydration synthesis. Now, dehydration synthesis, you're going to just break down the word. You're removing water and you're creating something bigger. You're synthesizing. So notice you're going to take OH and an H, an oxygen and hydrogen, and another hydrogen from that oxygen, and you're going to form H2O. And then because there's that oxygen left, that's going to connect, and you're going to have them, I jokingly refer to them as holding hands. That's an easy way to recognize a disaccharide. They're holding hands. There's an oxygen in the middle. It's never going to be these hydrogens are going to stay very stable. So it's always going to be that hydrogen attached to an oxygen. So here would be lactose. And then down here, this is sucrose. So what do these do? Well, disaccharides provide energy. We have lactose, which is found in milk. So mammals are going to be making that. And then we have sucrose, which is plant storage or transportation. Fructose is a monosaccharide. It should not be in this category, so my apologies about that. That should not be there. But that's going to be, sucrose is going to be for plant storage. Now we get into another category, oligosaccharides, and oligo is just many. It's just a shorter form. It's not a polysaccharide, which are hundreds and thousands of glucose molecules. So these might be 10, 20, 15 glucose molecules. They have a couple of different functions. In animals, oftentimes what they do is they form connective tissue, different types of connective tissue. They create gels, and they do that because they're adding polarity. So if we connect all of these together, and notice down here in we would have all of these little short chain. These are going to be the oligosaccharides, glucosamine. And what they do is they trap water here, and that creates a gel. So things like our cartilage or underneath our skin, those of you from anatomy class, you might recall I said this is what makes us squishy underneath of our skin. The other thing they do is they create a unique shape. So here's our picture of the cell membrane. I know we haven't gotten to it yet, but here's our surface. And these are all different types of glucose or fructose. These are all different types of monosaccharides. And you can see the fact that these form different puzzle pieces of shapes on the surface of the cell membrane, and that creates interactions of other cells. And then we end with our polysaccharides. And polysaccharides, the ones that we're really looking at, they're just repeating chains of glucose. The two types, we've got starch, which is going to be water-soluble, helical, that's storage form for plants. And we also have cellulose. Now, cellulose is not water-soluble. It doesn't mix well with water, and that's partly because of its shape, but it's really good at creating that plant cell wall. And what we see with plants, for instance, in the fall especially, what they're doing, these deciduous trees, they're going to be pulling all of their monosaccharides down from their leaves, and they're going to be transporting it down into their roots and then storing them as starch molecules down in the root cells. And then their plant cell walls that would have cellulose. Now, because they're just slightly different, they need different enzymes to break them down, to hydrolyze them. We don't have the enzymes needed for cellulose, but for instance, bacteria in cow stomachs do. So that's why cows can digest plant matter. And you can see the different starches on the left and cellulose is on the right. And it's just a matter of a slightly different bond, which creates different chemistry. And then down here we have the difference between animal polysaccharides. We have glycogen, and glycogen is this one on the right. There's really no difference between the structure they're all glucose molecules, but this one's highly branched. You can see it's just like they're coming off from the main stem there. This is how we store glucose molecules. It's found in the liver and the muscle tissue of animals, and it's very efficient energy storage for us. So here's just a quick picture of something that we learned about earlier. We're going to leave this here, and I want you to do 2 questions. I'm going to label something really quickly. I'm going to label this part, and then I'm just going to label this process. So this is going to be the first question, and then this process I'm going to label as number 2. Well, thank you, and we're going to be looking at these tomorrow in class.